Our cells are bustling metropolises. Within them, microscopic factories work tirelessly to fuel the functions of life. These are the mitochondria, the powerhouses of our cells. They generate the energy we need to breathe, think, and move. But what dictates the blueprints of these energy factories? The answer lies in a special set of genes found not in the cell's nucleus, but within the mitochondria themselves. Mitochondrial DNA. Mitochondrial DNA, or mtDNA, is a fascinating world of its own. Unlike the DNA in our cell's nucleus, which we inherit from both parents, mtDNA comes solely from our mothers. It's a fascinating echo of our maternal lineage, passed down through generations like a whispered secret. This unique inheritance pattern has profound implications for understanding human evolution and tracing our ancestry. But mtDNA is more than just an ancestral record. It's a vital instruction manual for the mitochondria. This DNA molecule is circular, a stark contrast to the linear structure of nuclear DNA. It contains genes essential for the production of proteins involved in cellular respiration, the process that converts food into usable energy. Without properly functioning mtDNA, our cells would be starved of power, unable to perform their essential duties. Imagine a city without power plants. That's the critical role mtDNA plays in our cells. Its genes hold the key to energy production, making it a central player in the symphony of life. In the next section, we'll delve deeper into the distinctions between mtDNA and nuclear DNA, exploring the unique characteristics that make the mitochondrial genome so fascinating. Our cells are like fortresses, harboring a central command center, the nucleus, which houses the majority of our DNA. This nuclear DNA is the master blueprint, containing genes that dictate a vast array of traits from our eye color to our predisposition to certain diseases. But nestled within the cytoplasm, the bustling city outside the nucleus, lie the mitochondria, each carrying their own set of genes, the mtDNA. While both nuclear DNA and mtDNA carry genetic instructions, they differ in significant ways. Nuclear DNA is a double helix, a long, twisted ladder with rungs made up of 4 different chemical bases. mtDNA, on the other hand, is circular, a continuous loop of genetic code. This difference in structure is just the tip of the iceberg. Nuclear DNA is inherited from both parents, a shuffling of the genetic deck that contributes to the diversity of life. This mixing of genes is absent in mtDNA inheritance. It follows a matrilineal line passed down from mother to child. This means that you carry the mtDNA of your mother, her mother, and so on, a direct genetic link to your maternal ancestors. The number of genes also varies greatly between the 2 types of DNA. Nuclear DNA boasts over 20,000 protein-coding genes, while mtDNA contains a mere 37. These genes, however, are absolutely critical. They are involved in the production of proteins essential for cellular respiration, the process that fuels our cells. Understanding the differences between nuclear DNA and mtDNA is crucial for unraveling the complexities of cellular function and inheritance. These 2 genomes, while distinct, work in concert to orchestrate the symphony of life. In the next section, we'll take a closer look at the fascinating inheritance patterns of mtDNA and their implications for understanding our ancestry. Imagine a family tree where one branch, the maternal line, carries a unique inheritance, a set of genes passed down unchanged from mother to child. This is the story of mitochondrial DNA, a genetic legacy passed down exclusively through the maternal line. Unlike nuclear DNA, which undergoes shuffling and recombination, mtDNA remains remarkably constant from generation to generation. The reason for this unique inheritance pattern lies in the very process of fertilization. When a sperm cell fuses with an egg, the sperm's mitochondria, located in its tail, are left behind. The developing embryo inherits its mitochondria, and thus its mtDNA, solely from the mother's egg. This makes mtDNA a powerful tool for tracing maternal lineages and understanding human migration patterns. Scientists can use mtDNA like a time machine, peering back through generations to trace the origins and migrations of populations. By comparing mtDNA sequences from different individuals and populations, researchers can reconstruct ancestral relationships and track the movement of humans across continents. This has revolutionized our understanding of human evolution and the interconnectedness of our species. The maternal inheritance of mtDNA also has implications for certain genetic disorders. Since mtDNA is responsible for essential cellular functions, mutations in these genes can lead to a variety of health problems. These disorders, known as mitochondrial diseases, are often debilitating and difficult to diagnose. Because mtDNA is passed down maternally, these diseases can affect multiple generations within a family following the maternal line of inheritance. In the next section, we'll explore the unique features of the mitochondrial genome, including its high copy number and susceptibility to mutations, and how these factors contribute to the development of mitochondrial diseases. Section 4. High copy, high stakes. Unique features of the mitochondrial genome. Imagine a library filled with thousands of copies of the same book. This is akin to the situation within our cells, where each mitochondrion contains multiple copies of its own DNA, the mtDNA. This high copy number is one of the defining features of the mitochondrial genome, setting it apart from the nuclear DNA, which exists as a single copy in each cell. The abundance of mtDNA is not without reason. Mitochondria are the powerhouses of our cells, responsible for producing the energy needed for all cellular functions. This high energy demand necessitates a large number of mitochondria and consequently a high copy number of mtDNA to ensure the smooth operation of these energy factories. However, this abundance comes with a price. mtDNA is more susceptible to mutations than nuclear DNA. Unlike nuclear DNA, which is protected within the nucleus and benefits from robust repair mechanisms, mtDNA is exposed to the harsh environment of the mitochondria, where free radicals generated during energy production can damage the DNA. Furthermore, mtDNA replication is not as accurate as nuclear DNA replication, increasing the chances of errors or mutations being introduced during the copying process. These mutations can accumulate over time and lead to a variety of health problems, particularly in tissues with high energy demands such as the brain, heart, and muscles. Another unique feature of mtDNA is heteroplasmy, a phenomenon where different copies of mtDNA within a single cell or individual can vary in their genetic sequence. This can arise due to the accumulation of mutations over time or inheritance of different mtDNA variants from the mother. Heteroplasmy adds another layer of complexity to the understanding of mitochondrial diseases, as the severity of symptoms can vary depending on the proportion of mutated mtDNA present in different tissues. In the next section, we'll delve into the world of mitochondrial diseases, exploring how mutations in mtDNA can disrupt cellular function and lead to a wide range of debilitating conditions. Section 5. When genes falter, mitochondrial DNA and disease. When the powerhouses of our cells malfunction, the consequences can be devastating. Mitochondrial diseases, a group of disorders caused by mutations in mtDNA, are a stark reminder of the crucial role these tiny organelles play in our health. These diseases are often multisystemic, affecting multiple organs and tissues, and can manifest at any age, from infancy to adulthood. One of the hallmarks of mitochondrial diseases is their wide range of symptoms, a reflection of the diverse roles mitochondria play in cellular function. Some common symptoms include muscle weakness, fatigue, vision and hearing loss, seizures, and neurological problems. The severity and specific combination of symptoms can vary greatly depending on the type and location of the mtDNA mutation, as well as the individual's genetic background and environmental factors. Let's take a closer look at two examples of mitochondrial diseases, MELAS and Kearns-Sayre syndrome. MELAS, which stands for mitochondrial encephalomyopathy, lactic acidosis, and stroke-like episodes, typically presents in childhood with stroke-like episodes, dementia, and lactic acidosis, a buildup of lactic acid in the blood. Kearns-Sayre syndrome, on the other hand, is a slowly progressive disorder that usually begins before the age of 20 and is characterized by progressive external ophthalmoplegia, which is weakness of the eye muscles, pigmentary retinopathy, or degeneration of the retina, and heart block. These are just two examples of the over 150 known mitochondrial diseases, each with its own set of challenges and complexities. The diagnosis of mitochondrial diseases is often a long and arduous journey, requiring a combination of clinical evaluation, genetic testing, and biochemical assays. The next section will delve deeper into the complexities of diagnosing these elusive disorders.